Welcome everyone to the Innovation and Quality Consortium's webinar series. We are part of the Translational ADME Leadership Group, and we are collaborating with the Clinical Pharmacology Leadership Group to present these two seminar series, one today and one tomorrow. We are working on novel modality discussions within this TALG and CPLG working group. Last year, we had spent some time talking about AAV-based gene therapies, and we split the two talks between risk assessments and bioanalysis, as well as the ClinPharm dose decision-making strategies. And following the same paradigm, we are here today to talk about cell therapies and immunogenicity risk assessments. And then tomorrow we'll talk about the bioanalysis for PK and ClinPharm dosing. So I'll go to the now to introduce the IQ's vision in the next slide. So the Novel Modalities Working Group, as I mentioned, is a combined collaborative group between translational ADME leadership and ClinPharm leadership. And it's being led by myself. I'm from Bristol Myers Squibb, and Nag Shamaturi from Takeda. And the mission for this leadership group or working group within the leadership groups are that we identify the risks associated with the novel next generation modalities. Our scope is cell-based therapies, viral vector-based therapies, and gene nucleic acid-based therapies. And once we identify the risks, we are proposing a risk-based strategy to drive the bioanalytical assessments for both preclinical IND-enabling studies and the studies for clinical development. The group discusses the recommendations and the best practices for novel bioanalytical and immunogenicity assays, best practices and standardization approaches for development and validation of these assays, and then use of these data sets for a roadmap for PK/PD analysis and dose translation strategies. So our seminar series are also based on this format. And because of the extent of the details and the content, we have to split them into two different seminars, and that's why we have one today and the other tomorrow. The three speakers who are going to cover today's talk will be Dr. Joachim Gockemeyer from BMS, who is a director in Discovery Biotherapeutics Group. Joachim has been at BMS for the last 15 years in different roles and responsibilities, mainly focused on biotherapeutic drug development. In the last 10 years, he has also focused on building a group for immunogenicity risk assessments and mitigations in the non-clinical stage of development. He received his training at the University of Groningen and the Dana-Farber Cancer Institute. Dr. Weifeng Xu is a director of regulated bioanalytics at Merck & Co. Weifeng has been in the field of immunogenicity for more than 10 years. He's a very active member in the AAPS neutralization antibody work group, as well as the European Bioanalysis Forum, NAB teams. He also co-leads the neutralizing antibody assay drug tolerance subteam at AAPS. After joining Merck at the end of 2018, Weifeng has led cell assay group within the preclinical development regulated bioanalytics to develop immunogenicity assays for both biologics, vaccines, and cell therapies. And lastly, Dr. Nanda Balasubramanian is a senior principal scientist in non-clinical disposition and bioanalysis group at BMS. He has 7+ years of immunoassay and bioanalytical experience. In his current role at BMS, he's supporting bioanalysis for CAR-T gene therapies and large molecules. And prior to that, he was the bioanalytical scientist role with increasing responsibilities at Pfizer and AstraZeneca, where he supported the oligonucleotide therapies, gene therapies, and large molecules. So I welcome all the three speakers today who will also be participating in the Q&A sessions. Thank you.
All right. Thank you so much, Viba. That was a great introduction. I would like to, my name is Jochem Geukenmeijer. As mentioned, I'm from Bristol Myers Squibb. And to introduce you guys, I really wanted to thank the IQ organization for organizing this webinar. And then secondly, I would really like to organize the people that you see on this slide here that have been the participants in these discussions that we've had over the last several months to come up with this webinar about cellular therapy. And really what I think the theme that we would like to highlight for this webinar is to give people an idea of, you know, what do we know right now about immunogenicity risk and bioanalytical challenges for cellular therapy, but also what don't we know yet, and then based on that, you know, what are the things that we think that we as an industry need to work on. All right, so first I'm going to talk to you guys through immunogenicity risk assessment. Then Nanda is going to walk us through the bioanalytical strategy and challenges. And then lastly, Wei Feng is going to talk about clinical relevance. And before I really get going, let me ask everybody that's on the call to mute their microphone, please. I think we have a little bit of background noise. All right, so I'm going to assume that most of us are familiar with CAR T therapy, but just in case, I wanted to give a quick introduction. So CAR T therapy on the left side, as you can see here, is really the modality where you would harvest the T cells from a patient that can then modify to express a chimeric receptor, and that's usually done with a viral system, as you can see in this figure here. And then those cells that express this chimeric receptor get expanded with the help of antibodies ex vivo, and then those CAR Ts get reinfused into the patients where they are geared up to interact with the chimeric receptor target that has been introduced, and in most cases that is a cancer cell, and therefore those activated CAR T cells can then destroy these tumor cells. And as you guys are aware, we have been working on this modality for a while now, and as you can see on the right side here, this therapy has shown some very remarkable efficacy in the clinic. So just a little bit more about the modality, just so we have a little bit of a framework on what we are talking about here. As you can see on the left side here, the cells, as I just introduced, as autologous T cells, we have obviously expanded on those where we can use T cells, we can use NK cells, we'll probably use other cells, but then there's also obviously the allogeneic option or the off-the-shelf option where people will use large donor banks of T cells or NK cells or a stem cell based modality. Then additionally, if we're talking about the different receptors, obviously there has been great progress being made in the design of the chimeric receptor, and that mostly is demonstrated by the first gen, second gen, third gen chimeric receptor constructs involving the intracellular activation domains that really convey the system, the activation of the T cell after binding of the antigen. But people have been also very active in the extracellular domain or portion of the chimeric receptor, which can either be, a single chain, and that's probably what we're going to talk most about, but that can also be exchanged for alternative scaffolds such as DARPins, nanobodies, but people can also just take a antigen or a protein that they would find useful to convey action. All right, so if we now move on to the immunogenicity risk assessment of these CAR Ts, obviously just purely theoretical. We know a lot about immunogenicity. We know most of the immunogenicity of therapeutics based on the large protein therapeutics that we have been working on for quite some time, and we do know that foreign proteins have the potential, and in most cases do, to some degree, elicit a immune reaction, resulting in anti-drug antibodies. And you can see that here in the bottom, where we think that, at least theoretically, the development of anti-drug antibody based on an adaptive humoral immune response should be a possibility for CAR-Ts. Additionally, since this is a intracellular expressed construct, there is the potential for non-human sequences to be presented on MHC class 1, resulting in a cellular adaptive immune response and resulting in CTLs or cytotoxic T cells that can play anti-modality function, thereby destroying potentially your CAR-Ts. And then lastly, as you can see at the top of this slide here, residual process proteins related to the production of the CAR-Ts, such as the viral proteins involved in transfecting the construct into the T cells, but also, potentially the antibodies used to expand the product, have the potential to be recognized, particularly by toll-like receptors, resulting in an innate immune activation, which then has the potential to crosstalk to the adaptive immune system and kind of change the balance of the immune system. So there is the potential for these modalities to elicit an immune response. So then, let's talk a little bit about impact of potential immune responses. Obviously, for us, the main concern is patient safety. And as we know from large molecule, particular monoclonal antibody immunogenicity, an immune response to your drug can really range from let's say nothing to all the way to from injection site reactions, neutralization, but also then anaphylaxis and very serious health considerations. Efficacy is obviously a concern. The destruction or neutralization of your CAR-T, either by destruction of the cell or neutralizing anti-drug antibodies, is a great concern for an immune reaction to a CAR-T. The chimeric autoantibody receptors, where the CAR-T consists of a endogenous antigenic protein, obviously moves us a little bit into the field of an endogenous protein therapeutic, where there's always the potential that an anti-drug antibody can cross-react with its endogenous counterpart, thereby neutralizing the function of that protein. And the example of EPO always come to the surface there. That is a concern. For the allogeneic CAR-T programs, obviously there's somewhat additional concerns related to graft versus host, or as some people have mentioned, it's really a host versus graft response to the off-the-shelf CAR-T. And that is obviously a concern, and at least we do have heard some reports that we think that allogenic CAR-Ts, due to their shorter persistence in the system, are more likely to be redosed, and obviously redosing of the same modality increases the risk that your immune response will react. And then lastly, expression of endogenous cytokines to further enhance the activity of the CAR-Ts relates to the same risk as mentioned for the autoantibody receptors, where you express a endogenous protein. Okay, so what have we seen so far what do we know about immunogenicity? Obviously this is a fairly new modality, so we don't know that much, but we do have, we have 6 approved products in the clinic right now, and we do have immunogenicity data for those programs. As you can see here, we do see various ranges of anti-drug antibodies for these products. And then we kind of split it out as in what do we know ADA or anti-drug antibody wise, and what do we know for cellular immunogenicity response. We've seen some anti-drug antibodies, as you can see here, although as noted on the right there, none of these have shown an impact on expansion, persistence, or efficacy of these products. Most of these products have looked very well into the potential of cellular immunogenicity. So there is not that much that we can say about that, although I think as most of us probably know, looking through the literature and the programs that are in development right now, there have been reports of cellular responses. There have been reports of ADA responses to CAR Ts that do have some impact on the persistence and efficacy of those products. Okay, so if we look at immunogenicity that is inherent into the construct, if you look on the right here, where we've made this little cartoon of the basic structure of a CAR T with a single chain, extracellular, some activation domains, intercellular, fairly similar to what we do for looking at large molecules. The immunogenicity risk is related to the non-self or non-human sequences pertained in this construct. For these constructs, kind of pertains to the CDRs for the single chain. Those can be for people that didn't make those CDRs can be recognized as foreign sequence. Obviously, linkers can be non-self, and then the junctions between different domains always have the potential to be generating novel non-human sequence. So those are all containing the immunogenicity of the sequence within the receptor construct. As mentioned, the residual production-related proteins, there's a good amount of proteins involved in making these cells and in expanding them. So the viral factors used to transfect the cells, as we know, viral factors can have viral proteins can be immunogenic. The expansion antibodies, potential CRISPR or TALEN related proteins used to delete genes within the CAR T all convey some kind of immunogenicity risk. And then obviously, as mentioned, the allogeneic CAR Ts, the potential for graft versus host due to MHC mismatch or TCR recognition is a potential immunogenicity risk if one would leave those in place. Next here. So usually when we like to look at immunogenicity risk, we like to consider the purely inherent risk related to the construct or the modality or the sequence, and then obviously there's patient and disease-related immunogenicity risk that kind of modulate that risk, and obviously we need to take those into account. As we've seen so far, most of the products that are on the market right now are oncology projects for wet tumors or B-cell malignancies that are obviously immune suppressive. On top of that, a lot of these products have a lymphodepletion step that further inhibits the immune system to mount an immune response, thereby therefore the risk is therefore somewhat decreased. If we start expanding our indications and move to solid tumors, or we move into non-oncology settings or potentially immunology settings where the immune system is considered to be more intact, more active, or in the case of immunology might be overactive, obviously it is easier for the patient then to mount an immune response, and we have seen that from other modalities too. And then obviously there's the patient factors where, as mentioned above, the general status of the immune system is a risk factor or a decreased risk factor. And then also, how much pre-existing immunogenicity is there, particularly to the residual proteins. We know particularly for AAV or Cas9, there's a fairly large percentage of the general population that have pre-existing antibodies to those proteins. And then lastly, we did want to talk about serial treatment, or particularly serial treatment with different products that are CAR-Ts, mostly related to the fact that large portions of the CAR-T are fairly exchangeable between different therapeutics, particularly the intracellular portions. And therefore, there's always the risk that if you have been treated with therapeutic A and a patient will move on to a different product, once there is an immune response to one portion of the chimeric receptor, if that portion is also present in the next CAR-T, that pre-existing immunogenicity would then translate to that new product, and obviously that is then a concern for those patients that would have a follow-up or serial CAR-T treatment. Good. So a little bit about immunogenicity risk assessment, and mostly preclinical immunogenicity risk assessment. The tools that we have in hand right now are mostly just, they're really adapted. They're fairly similar tools that we have developed for large biologics, such as mostly monoclonal antibodies. And what we have done is we've really, tweaked those tools to accommodate these cellular therapeutics. And so those tools are really the in silico tools that most of you are probably familiar with, where you can just scan through a sequence and look for MHC class 2 potential binding sites. And what we have done for these cellular therapies, since we know that the construct is expressed intracellular, so we have concerns about class 1 and class 2. Luckily for us working in this field, a lot of these in silico tools accommodate class 1 and class 2, and therefore we can scan fairly straightforward through our sequences. The in vitro assays, so the T cell proliferation assays, where one uses a large donor set to see if foreign or potentially therapeutic proteins have the potential to elicit a proliferative response, can be used, although one needs to get their hands on a recombinantly expressed portions of the chimeric receptor, which in most cases then people use the extracellular single chain to see if that has any potential to elicit T cell proliferation. MAPS assays have over the last several years become fairly popular for monoclonal antibodies. We can modify those assays and actually use on the actual drug product and see if we can elute processed and presented peptides of MHC and MHC class 1 and class 2 molecules, and sequence those to get a better idea of what peptides from a chimeric receptor are actually processed and presented on those receptors. And then lastly, the innate activation assays that can be used for to look for innate immune system to be activated are fairly well described as whole blood or PBMC based assays, where with a cytokine readout to see how much innate activation those potential products elicit. Let me do a little time check here. Okay. So mitigation strategy, that's always a good thing to have in your back pocket once we start looking at the immunogenicity risk. So obviously, based on the risk factors that we identified in the sequence, using those tools that we just talked to, and the knowledge that it's mostly the CDRs and the linkers and the domain junctions, we can play with that. We can select single chains with inherently low in vitro and in silico predicted immunogenicity risk. We can look at our domain junctions, and we have the potential to move those a few amino acids one way or the other, and see if that would lower the potential risk. And so that falls all under the protein engineering strategies that can be deployed during the phase of your development of your product, at which we have the flexibility to pick a lead molecule or receptor. And the allogeneic CAR-Ts obviously are making wide use of CRISPR technology, particularly to delete the TCR and the HLA and potentially the CD52 molecules of their allogeneic CAR-Ts, thereby greatly reducing the risk of developing graft versus host disease. And some groups have also experimented with expressing some more tolerogenic receptors on the surface to minimize risk. And then obviously for the process-related impurities, these are highly individualized products. Therefore, they're monitored on an individual basis. There's WHO standards that can be deployed to look at how much of those residual proteins do we have and do we allow in our products. And then obviously based on in vitro assays, we can do some work on seeing at what levels do we think that these impurities pose an immunogenicity risk. Okay, so then lastly to the question of what do we know, what do we don't know, what would we like to work on? In general, the data that we have and obviously the novelty of these modalities means that we don't really have that much data. So there's a great need for us to get more data, to get a greater understanding of the mechanisms that drive potential immunogenicity in the clinic, to also figure out the tools that we have right now, how predictive are they? Do we have any correlation? Do we need to tweak those? Do we need to work on those? And obviously to get that, we need more reverse translational research. So we really need to get more data published. We need to get more data shared. We need to get more data from the clinic. We need detailed information on, do we get anti-drug antibodies? Do we get a cellular immunogenicity response? What's the HLA type of those patients that develop the response? And how does that correlate back to what we predicted preclinically with the tools that we have? And how much impact do we see of a potential of an immune response? And was that predictable in one way or the other? And that really is I think the way that we are going to the efficiency of our tools that we have in place. All right, so that was it for my section on immunogenicity, and Nanda is going to walk us now through the bioanalytical strategy and challenges.
Hello, everyone.
Do you want me to move the slides, or?
Yes, please.
Sure.
Thank you. Thank you, Joachim, for a very wonderful outline of the immunogenicity aspects here. My portion of this presentation will focus on the bioanalytical strategy and what's being done by various companies, and our recommendations in terms of the immunogenicity assays that we have in hand. There are what gaps to address, and I also touch upon the PK assays that we have. So next slide, please. So as Joachim alluded to, cell therapies are capable of inducing both humoral and cellular immunogenicity. The overall strategy is very similar to large molecules, and large molecule guidelines are applicable as long as we are implementing an ligand binding approach. And in general, autologous T cells pose low to medium immunogenicity risk. Having said that, there should be a clear communication that with the health authorities and understanding of what will be measured at what stage. The critical attributes to consider are impact on exposure and expansion, and also the patient and product related attributes of your product. For humoral immunogenicity, you would use a tiered-based approach for LBA. LBA assay for ADA is one of the initial assays that would be implemented, that's usually implemented. And we have a clear guidance and consensus based on the large molecule of what are the expectations of the ADA assay should be. But there are situations where, you may need a cell-based FACS assay to detect antibodies to CAR T, where reagent availability could be an issue. So you should want, based on the mechanism of action and reagent availability, there will be a requirement to have a cell-based ADA assay or a immunogenicity assay. NAB assay requirement, it's still not completely clear. If requested, mostly it will be implemented in phase 3. And here also you could discuss the benefits between a competitive ligand binding assay versus a cell-based assay. Lastly, cellular immunogenicity, ELISpot and fluorospot are widely used. Also, based on the stress because of the sampling volume required, alternate assays may be needed to be considered for this aspect. So that, to cover the whole immunogenicity perspective, one can implement a dozen or more assays, but we have tried to focus this talk on the very, the core assays that would be needed. Next slide, please. So for humoral immunogenicity, as I said earlier, the anti-CAR antibody assay usually would be implemented during the FAH start. And if you have the reagents available for an LBA, you can use this tiered approach that's generally used for large molecules. The assays could utilize purified reagents or cells that express the target. But when soluble ECD domain or the extracellular domain is available, a bridging ligand binding assay would be sufficient, as indicated in the diagram on the right. So in these cases, drug interference and matrix interference are critical, but how to address these questions, interference is still, we don't have a clear path on how to go about this. On the alternate way, a cell-based assay format is appropriate when you don't have, when your extracellular domain has solubility issues. Also, you have risk of masking immunogenic epitopes during labeling, and you may need to monitor immunogenicity for the entire CAR based on your CAR sequence. So that's a situation that requires a cell-based assay. And the cell-based formats could range from a plate-based assay where your cell line expressing CAR or cell membrane is bound and then bound to a plate or immobilized. Alternatively, you could use a flow cytometry-based assay using recombinant cell line expressing your CAR. So these are the two approaches you could implement for FACS-based ADA assay. Next slide, please. So for the ADA assay formats, what I've done here is to compare the various formats and also go through the challenges that you might face. The advantages of an LBA-based assay are, based on our large molecule experience. It's easy to implement, and when appropriate reagents are available, standard sampling, collection, and processing would be applicable. But the challenges would be to have those reagents available, and as I said before, the matrix and drug interference could be an issue. On the right is a summary from Novartis in terms of the ADA domains and features that are detected in their various approved, CAR-Ts, and you could see the cell-based assay would cover a more robust robust coverage of all the CAR, while the soluble assays would cover portions of, or only the external exposed domains of the CAR. If you go to the next slide. So here, the advantages of the cell-based assay are once you transduce cell lines, you have higher transduction rate, consistent CAR expression for a long term. No labeling requirements for CAR proteins, and solubility is not a concern. And this enables CAR presentation in its native state with any potential post-translational modification. But having said that, there are multiple challenges for cell-based AD assay, including the availability of the pertinent cell population. Adaptation of cut point would be an issue. Loss of CAR expression and population shift over a period of time will be a high concern, and high background potential for non-CAR specific responses has been observed in some cases, and wild type T cells may be needed as controls throughout your course of the study. And this would kind of complicate your sample collection process, and it would require specialized labs, because not all CROs, if you're implementing a fax-based approach, not all CROs are capable of doing it. So it adds a layer of complexity to your immunogenicity assessment. The next slide, please. So here, what we have said, try to address is show the formats of the assays that you could use from a ADA perspective, or a solid-based ADA assay, where when you have a soluble target and the pertinent reagents, a general ADA assay that's implemented for large molecules could be used very easily. And a cell-based approach could be used, where you would have a cell line expressing CAR receptor and a target-induced reporter readout. So the readout would be a soluble target, if you have a soluble target, but you might need a second cell line depending on your mode of action, or if the protein needs to be expressed in a transmembrane protein that needs to be expressed in a cell line. So you would need a second cell line for a cell-based assay. So you could have, if you have the cell-based assay, you could implement that based on the mode of action. Alternatively, one could take advantage of the potency assay, or the cell killing assays from the CMC teams, where you have the potency assays available, and you could be repurposed that for a NAB assay. If you could go back one slide. Sorry, yeah, go forward, please. Sorry, I thought I'm missing something. And this, these assays could be used for as using a second cell line. You could implement those readouts as well in your ADA assay. So you essentially have 3 different formats that could be used for your ADA assays. Next slide. So the challenges that I've listed here are that we have discussed multiple times. The availability of the soluble CAR reagents has been an issue for many programs. Masking of domains during labeling is a concern. In terms of the cell-based assay, the availability of patient-specific T cells is not easy. The time needed for generation and expansion of CAR expressing cell lines should be taken into account when you're planning your assays and the timeline surrounding that. A loss of CAR expression and a population shift is a concern, and one needs to be prepared for that and have a large batch of cells frozen ahead of time when you create those initial lines and expand them. High background potential for non-specific CAR sequences could pose issue, and wild-type T cells should be included in your, as a controls during the process. Next slide, please. So moving on to the cellular immunogenicity aspect, this is a depiction of how you have a CD4 activation or CD8 activation. You can click one more time, Joachim. Thank you. So the short peptides, usually the 9 to 11 mers, is expected to activate your CD4 pathway. And if you click one more time, the longer peptides would activate the CD4 pathway, usually 12 to 20 mers. So having, instead of having a two group of peptides, the best approach that that's been generally implemented is in something in between. If you can click one more time, is 15-mer peptides. So this enables activation of both MHC class 1 and class 2. It's a good optimal compromise between both the pathways, and it activates pretty well the both CD8 and CD4. This also gives you a ability to characterize, if you want to, in terms of using short peptides, if you want to understand your cellular immunogenicity in depth. But the best approach to start with would be to have a 15-mer peptides, enables both activation of MHC class 1 and class 2. In general, ELISpot and fluorospot assays are widely used for this. And one more time. And I'll just touch upon, before going into the ELISpot, the idea here is to discuss how the critical aspect of this assay for cellular immunogenicity is the peptide design, because as you saw in the previous slide, those will be your positive controls in a way. So you need to make sure you're having the right antigen in there to stimulate your PBMCs. So the general consensus in the approach for this path, for these assays, is to use 15 mers, as I talked about in the earlier slide, but also to ensure coverage of your entirety of the protein or the CAR construct. So you would have a 4 amino acid walkover with a 11 mer overlap. That would be the best way to cover your entirety of the protein, and also to be able to have manageable number of peptides in your pool. The second aspect of this would be the peptide pooling. If you can click one more time. So one can be innovative in terms of how you would pool based on the extracellular domains. You could be pooled in one peptide pool, while there are others could be the transmembrane and the internal intracellular domains could be in a different pool. So one could be innovative in how you would pool your peptides so that you could characterize your cellular immunogenicity in more detail. Next slide, please. One critical aspect here I forgot to mention is to ensure that the peptides are capped so that you don't have partial peptides in there. So the general outlay of the ELISpot assay everybody has seen here should be familiar with this. So the two aspects that wanted to cover, one is the effective T cells on the left, and one can incubate the PBMCs with the antigen for a period of time, also to measure memory T cells. The attributes and challenges for this ELISpot assay would include, PBMC isolated from patient and stimulated with peptides corresponding to the CAR. That would, the PBMC isolation is critical, and this requires a high blood volume, and sampling time should be determined in context of lymphodepletion. So given the complexity of this assay, and currently there are not much guidelines for validation parameters, so one could argue if it should be a validated assay or a fit for purpose, exploratory assay. And also non-availability of patient PBMCs makes it even more tougher for you to validate, and you're going to use appropriate positive controls for your method qualification or validation. For everybody who's worked on this assays, understand that sample transport is a pain, and assay implementation and cost also should be considered. So all these aspects make implementing cellular immunogenicity, especially an ELISpot assay, as highly painful process, and that necessitates the need for autologous methods like a Luminex assay or a chromium release assay that could be used in conjunction with it or independent of it. It's still not being, it's still theoretical, but that's something that the industry should be looking into, because that will add more value for the cellular immunogenicity. Thank you. So in general, so far what we have discussed is the potential inhibitory mechanisms of both humoral and cellular immunogenicity of CAR-Ts. So a neutralizing antibody could impact the target cell binding, and in a way, the same could be the situation with the cellular immunogenicity as well. So what should be considered in, the important aspect to be considered here, if you click one more time, is how this immunogenicity impacts the exposure and expansion and the persistence of CAR T cells. So having these assays in place should be also looked in a holistic way in terms of exposure and expansion of CAR T cells. So I'm going to touch upon the cellular kinetic assays. Next slide, please. Sorry. So the general features of CAR-T cellular kinetic assays are, the important aspect here is to understand that the cells expand in patients upon target engagement and can persist for years. So when you look at this from a small or large molecule perspective, these cells can proliferate. So that would have an impact on your cellular kinetic profile. And it has two distinct phases of contraction and persistence. And we have seen recent reports where some of these patients had these cells persist for about 10 years. And variability exists from patient to patient, and dose exposure relationship are not strong. The graph in the left shows the, sorry, please go back to the previous slide. So the graph in the left shows the Tmax about 10 to 14 days. It has a characteristic expansion phase and the and the persistent phase. You have exposure of 0 to 28-day AUC. That's the majority of the total exposure, and the persistence may be upwards of 1 year or even greater. And the matrix used here would be blood and bone marrow. The graphs in the right are to indicate that the necessity to set up a right sampling plan based on what you're looking to measure. So on the x-axis, you have, on the middle graph, you have 0, 10, 20, and 30 days. So these would help you characterize the dip and the redistribution and the rapid expansion phase. But if you don't have that sampling period, you might miss those phases. So it's critical to understand the sampling plan and what you're measuring when you're considering the cellular kinetic profile. Next slide, please. So there are two ways of measuring this that's been implemented. It generally, the polymerase chain reaction that quantify the CAR-T transgene indirectly is the qPCR method, where the key reagents being the primers, probes, and the target sequence, which are usually in plasmid or cell line. The assay details include you need a genomic DNA, normalization to a reference gene is required, and the sensitivity is around 50 copies per microgram. And you do not have concerns of ADA interference. Sorry, 1 second. The samples are stable and frozen after collection, so this method has some advantages, and the DNA extraction process is well established, and it's easily implementable across multiple CROs. So this could be the go-to method for your CK method. But alternative method where you don't need to have a normalization process, and the cell surface antigen could be used to is the FACS method. The antibody to cell surface antigen and the control cell lines are the key reagents here. So the FACS method has its own advantages, but the sensitivity could vary, and the usefulness to understand distribution of CAR expression over a period of time makes this method advantageous. Suspension of PBMCs should be analyzed fresh, so that puts a strain on the sampling process. Implementation requires specialized analysts, so it's not that easy to implement compared to that of a qPCR assay, and potential for ADA interference does occur. So these two assays have its own merits, and it could be implemented independently, or it could be a combination of both, and that has been done for different approved drugs. Next slide, please. One critical aspect that we wanted to touch upon here is the primer and probe design for the qPCR assay. So the single copy gene as the reference gene is needed to normalize the genomic DNA input. And you would have target conserved DNA sequences between species to enable cross-species comparison in your preclinical studies. Also, you want to ensure the amplification efficiency between the target and the reference gene is very similar. And one critical aspect of your primer design should also take into account in situations where you're redosing with different product, it may be necessary to differentiate between two CAR-T products. It's still, for the upcoming programs, but it's still something you need to consider to enable this differentiation when you're taking into account your primer and probe design. The average copy number from cell product is known, but the cells may have different copy numbers, expand differentially. So that should be also taken into account when you're trying to correlate between the amplification into cell number. So it might not be 100% directly translatable. Next slide, please. So in summary, what we have looked at is CAR-T modalities, complex and presents a variety of complex bioanalytical challenges. The CK methods, one could implement flow, which is a direct method, or the indirect PCR method, or could a combination of both. A risk-based tiered implementation of immunogenicity assays would be applicable when you have the LBA method implemented early, and the cell-based methods could come in later. Both humoral and cellular immunogenicity needs to be developed. Humoral assays, yes, validated. Cellular immunogenicity assay validation is still a question of the depth of attributes that you need to characterize for cellular is limited. Cell-based assays provide distinct advantages, as we have discussed, but LBA could be advantageous in certain scenarios. Consistent interaction, not only with the health authorities to establish the expectations, but also a collaboration within your team with discovery and CMC teams is important to be able to consistently source these reagents that are needed to support various assays. So thank you. I'll give the stage to Wei Feng for his clinical part.
Can you see my screen now?
Yes, but can you put it in presentation? Okay, thanks.
Oops. Okay, just a time check. Yeah, I should be able to finish in 15 minutes, so we will have enough time for the discussion. Yeah, and thanks for the team. I'm actually relatively new to this therapy field, so most of the things I'm going to share today, it's actually the all this discussion from the team. And the first thing I'm listed here is again the 6 approved CAR-T, right? But I want to direct your attention to the top 2, Kimaro and Yescarta. And you can see they actually have a very similar SCFV structure, right? But once using cell-based assay and once using the bridging assay, and you can actually see a very different in terms of the ADA positivity, right? And this could be related to either a cell-based assay or the bridging assay. So sometimes the assay format indeed will impact a lot. That's why Ananda mentioned we actually need a wild type T cell as a control in order to dig deeper, right? The other important thing, as you can mention, it's all these anti-CAR antibodies, they actually showed no impact on the clinical efficacy of all this approved CAR-T. And why that is the case, right? It could have 3 explanations. And it's also interesting that an anti-CAR antibody are only seen after 3 months after dosing for all approved CAR-Ts. And the 3 reasons for the no impact and this delayed anti-CAR antibody response could be, first of all, of course, the immunodepletion, right? So if there's an immunodepletion, then your B cell counterpart will be eliminated. And that will definitely require longer time for B cell to recover and amount an anti-CAR antibody, right? So the first question is, it's a lymph depletion always done? Obviously not, definitely not in all the solid tumors. And then even if we have, how complete is the depletion, right? It would be interesting to see the completeness of the lymph depletion and the relationship to the anti-CAR antibody response. And the second thing, it's related to the target indications, right? For all these 6 approved target product, 4 are targeting CD19 and 2 are targeting BCMA. So all of them are actually targeting to depleted B cells. The cells produce antibody. And the last one could be interesting, right? So the ADA might be absorbed to the CAR-T at early stage when ADA level is still low and the CAR-T is plenty, right? It's almost like a drug interference. So you have much lower circulating anti-CAR antibody in the circulation. And for the clinical relevance for the NAB against CAR, first of all, there's none of the approved product mentions the NAB in the label, right? So currently, most of the NAB assets really for the exploratory, and is NAB really needed if PK is already impacted, right? We're going to touch that in the later slides. And also, we know from our team members, and some of them were actually requested by FDA, even with the lower ADA rate and then no much impact on the cellular kinetics, right? So probably we needed to be kind of thinking the NAB assay early on. And of course, as you can mention, the NAB testing could be more relevant if there's a repeated or dosing with the similar CAR constructs, right? Then the host will have increased opportunity to see the antigen to mount the NAB response. And of course, we can use a similar strategy as a biologics. So we can use the NAB positive versus NAB negative groups to analyze the impact on the efficacy, and this definitely will need a NAB assay. And the 2 other things I want to share here is we definitely see some new trends in the biologics NAB field, right? First of all, is the ever since the 2019 the FDA new guidance for the biologics, and it actually mentioned the first time PK/PD and ADA could replace the NAB testing. And we already starting to hear some interesting case study around, including my group, right? So, and the second trend is previously FDA always wants you to have a NAB assay for the biologics, even with the project with a very low ADA positive rate, right? But recently I heard directly from some FDA officer that now they actually kind of tended to agree for some of the low ADA positive rate projects, you probably don't need a NAB assay. So both of this trend could be included in the cell therapy area. And regarding the anti-CAR antibody and the impact on the efficacy against the solid tumor, I picked 4 case studies here because each one of them actually had some very interesting observation, right? The first one is targeting alpha folate receptor. And you can see they definitely detect the antibody against the CAR-T, and it has been shown to be associated with reduced CAR-T activity. And their concurrence may have led to the rapid clearance of the CAR-T, right? And also for the second case, it's a classic one against the TAC72. And they actually detect a NAB against the SCFB, and they thought it's associated with elimination of the CAR-T. And of course, I want to point out that these are just association, right? It may not be the cause and effect. For example, the cellular response may both contribute to the rapid clearance or the elimination of the CAR-T, right? We just don't know without that data. And the third one is actually targeting the carcass on RCC, and they have a very interesting observation here as well. They see very high incidence of the anti-chimeric antibody, but as high as more than 80%, right? But if you use the parental monoclonal antibody alone, that monoclonal antibody actually has a very low, much lower anti-ADA. So this suggested the high immunogenicity could happen when we put the monoclonal antibody and convert it into SCFE on the cell surface, right? And they also found that this ADA can inhibit the cytotoxic activity of the CAR-T. So probably it has a NAB population in there, right? And for the last one, it's actually using a newer generation of the CAR-T. It's the FRP5 SFE against the HER2 positive sarcoma and the glioblastoma, right? And their observation is the core transgene can be detected up to 2 years. So the persistency actually is pretty good, right? But the clinical response will actually unsatisfactory. So that means the good persistence not always will lead to a good response, and the NAB might be play something here, right? And the main conclusion, as you can mention, is pretty much for most of the approved and also on ongoing clinical trial or the publication, we're not seeing a very intensive investigation for both the cellular and the humoral immune response, right? And for the solid tumor, the lymph depletion can also be helpful to reduce CAR-T immunogenicity and helpful for the efficacy. For the clinical observation of the cellular immune response, I also picked the 2 cases, which are both pretty classical. The first one is against the CD19C20, and they used the PBMC chromium 51 releasing assay, so it's essentially a killing assay, right? And they actually found that the cytotoxic T cell response is targeting against the neomycin, which is a selection marker, as well as the high TK, which is the suicide domain, right? And for the second case, it's targeting the CAIX on RCC again. And they found the cytotoxic cellular reactivity was directed against mostly the CAR protein, but it can also direct against the retroviral epitope. So you see, just by these 2 cases, you can see the cellular response can against any of the non-self sequence, right? So that makes that could make the cell therapy quite complicated in terms of looking into the cellular immune response, especially if you have a multiple layer of the engineering, for example, the using CRISP to knock down, right? And so far, only one approved CAR-T product has label information for cellular response, and it shows no impact. And we know FDA has requested cellular assays, and we don't know the detail about that CAR-T, right? And also the lymph depletion may reduce immunogenicity for both humoral and the cellular immune response. However, we have to be careful about the lymph depletion, as the newest FDA guidance, which just came out last month, right, says the repeated lymph depletion has to be careful because it may pose life-threatening risk of the myeloablation to subjects. And what is the clinical relevance of the cellular response data, right? That's also an interesting question to ask, and it may not be the main driving factor for the decision making, right? It might be helpful to mitigate the immunogenicity, especially cellular response, for the next generation of the CAR. And as you can mention, the sample collection and the testing is challenging. So we need a really good logistic for sample collection, shipment, and storage. And the last one, it's the change in the transfection vectors could reduce the cellular immunogenicity, right? So in some cases, if you have the stable transfection, then they becomes a part of the cells that can always induce the cellular immune response potentially. And this slide I borrowed from our team member Sida, right? I think she's in the audience. So this slide I just wanted to show you again the complexity of the current and the future CAR therapy, right? It can have like 5 different layers of the engineering on the left side, and on the right side you can see all different immune response against the CAR T, right? Including both humoral and the cellular. And for the cellular, it could be T cell and NK cell and other innate cells. And because of this complexity, right, if we wanted to have all the assays for all the components, basically we will need more than 20 assays for exhaustive immunogenicity assessments, right? I just listed some of the them here, the CAR, of course, and the amylin molecule. If we are talking about the IL-15, right, then because this is a little different from biologics. For example, the biologics IL-2, usually it will have some like a mutation, or it will have some conjugation, right? But for this, in this case, for the IL-15 in the CAR-NK, it's pretty much like endogenous, right? So we don't expect a high risk of the immunogenicity. So in that sense, the initial ADA assay might be enough, and the NAB can really depend on the ADA data. And also for the suicide safety switch, it probably would be dependent on whether it's on the surface or in the intracellular, right? If it's on the surface, then more worry about the humoral immunogenicity, and if it's intracellular, then the cellular immune response should be considered more, right? So with all this consideration and assessment, the good thing is there's a 2015 FDA guidance, right? So it says, first of all, if immunogenicity is a concern, of course it will be the concern for every component. But the good thing is if adequate assays are not yet available, baseline post-treatment blood and plasma is appropriate, should be preserved for later evaluation, right? So once assays have been developed. So there's indeed some flexibility here. And again, 4 bullets I found quite to be quite interesting and related to the immunogenicity topic today, right? The first of all is the newest guidance says we should include an assessment of any impact that and these additional elements will have on CAR T cell specificity, functionality, and immunogenicity, and of course safety, right? And here it mentioned the transgene sequence that are unnecessary for the biological function of a product that may be immunogenic, right? So it's the best as a general guiding principle. We should avoid this unnecessary transgenes as much as possible to reduce the immunogenicity. And again, the previous administered CAR T in the starting material may impact the safety, efficacy, and the immunogenicity. And in the section, in the last section of this new guidance, it describes a lot of the manufacturing change, right? For example, the vector change, the growth factor, both different growth factor or even the concentration for the expansion, and all this could be cause changes in the immunogenicity, right? So we should really take notes for all this manufacturing change and to see whether it can be traced, be collected with the immunogenicity data. So as everybody mentioned, right, there's a lot of unknown and uncertainty regarding the immunogenicity strategy for the CAR-T. So here just on some rough thoughts. And of course, we must do the immunogenicity assessment and mitigation, and for every components, right? So we needed to consider all the sequence necessary, and we needed to prioritize based on the immunogenicity risk. And easiest thing just to start with the ADA assay against the core portion, right? And one of the things which could be very unique for the therapy, at least at the autologous stage, right? It's a highly personalized and case by case. So there's a lot of interest from clinical or clinicians. For example, they will be very keen to know the cellular kinetics, the expansion, the persistence, and the phenotyping of these cells in vivo, right? What's their fate, and do they change their phenotyping? And of course, the all this related to the efficacy, right? And there's a very new paper came from Carl Jung's group. They monitored 3 patients almost like a decade ago, right? Who were among the earliest to receive the CAR-T therapy, and they actually find as of now, 10 years later, they can still detect this CAR-T, and the phenotyping analysis is it's amazing. So with this kind of collaboration with clinician, maybe we can also get a lot of useful data for our immunogenicity strategy, right? And of course, we always needed to have a more clear communication with health authorities, right? We can have a pre-IND discussion, and once we get the initial phase 1 data, and based on this data, we can further fine-tune our strategy. For example, if both persistence and efficacy are good, then there's probably no needs for both NAB and the cellular immune response, right? If there's a low persistence, then it may due to either cellular response or the NAB. And if persistence is still there but no efficacy, then most likely it could be due to the NAB, right? But again, what are we going to use this NAB or the cellular response data, right? What a clinical information and value it could provide to the current CAR-T or the future generation of the CAR-T, right? So all these are the questions we're going to continuously discussing and fine-tune with the new data. So as a conclusion, and definitely we see the cellular therapy have been shown to have a remarkable efficacy, especially for the blood tumor area, right? And the immunogenicity bioanalytical strategies in place and being developed. And there's still a lot of unknown remain regarding many questions and challenges. And best practice need to be developed within industry, and of course the continued communication with the regulatory agency, right? And we're looking for the white paper in this field, so we will have much clearer guidance. And then I will stop there.
Well, thank you, Wei Feng, and thank you to Nanda and Joachim also for today's the 3 aspects of the talks related to risk assessments and bioanalytical strategies, and also bringing the clinical relevance. So we have about 8 minutes, and I want to quickly start talking about some of the questions, and I've already assigned them. So what we can do is go through some of them. Joachim, we can start with you first and look at the questions that were that came up for you, and then go to Nanda after this. So I think before we start, there's a question about will we get the slide deck, and I think yes, we are okay to provide the slide deck. It may be sent later to all the people who registered, and I'll let the organizers confirm that. And so. Okay, so the first question is from Dr. Amy Rosenberg, and this is about, are there examples of ADA to CAR T cross-reacting on endogenous proteins? And Shokam, do you want to give it a try to answer?
Sure. Amy, I think it's a very good question. I think at least for what we have looked, I'm going to first start out saying that we haven't seen any data of these CAR Ts that have a potential antigenic protein as part of the CAR T. So I think to a degree, this is mostly a theoretical question for now until these products will generate some more clinical data, at which point we will probably see whether this is a risk or not. And then I think the additional note that I would make is, you know, the immunogenicity for endogenous proteins that we have seen are all recombinantly expressed, you know, by non-human cell lines, and therefore we know that there are slight differences in glycosylation, in folding, etc. So it might be a very different situation where these endogenous proteins are actually expressed by the patient's cell themselves. So I still maintain that there's probably some risk, and we probably should carefully look at it for that potential of cross-reactivity, but it also might be a different situation than we have seen for the endogenous protein-based therapies that we have currently clinical data on.
All right, just so that I can cover all these questions, the next one is from Dr. Shobha Purushottam. Is the patient's immune status a factor in the risk assessment? Shokam. Sure.
Yes, I think as we had mentioned, obviously the general status of the immune system is likely to play a big role. If you have a severely compromised immune system, either due to previous treatment or due to lymphodepletion or the target that the CAR T addresses, and thereby, as mentioned, depletes your B cell. In short, if you don't have much of an immune system, you're not going to be able to mount much of an immune response, or at least greatly delay that. So I do think that is some risk factor that one should take into account in the risk assessment.
Great. Wei-Fang, a question for you. In regards to the comment about FDA requesting NAB assay use even with low levels of ADA, is there any indication if they should be considered in phase 1 trials or added to later stage trials in these type of therapies? Question comes from Amanda Goldston.
Oh yeah, if it's about the phase, then we know the NAB assay for the biologics usually it's only required for the phase 3, right? So, but we also have some like a high risk, for example, recombinant protein with endogenous counterpart. This would be the phase 1, right? And if it's a phase 1, then I guess you have no choice. You probably wanted to have prepare your NAB assay together with the ADA assay to be prepared for the phase 1, right? For the phase 3, you can wait until you see the ADA data.
All right, 2 questions for Nanda. One is from Dr. Shobha Purushottam again. It's about cut point setting as a recommendation. Is it still 50 individuals, or do we typically have a certain number of patients in the trials? How do you address cut points for CAR T's assays?
So, depending on the assay format you're using, if you would use normal individuals in this case, samples available from the subjects would be very limited. So you wouldn't be able to use disease population in this scenario.
But would you validate with normal and then just do more of an assessment with the study specific later?
Yes, yeah, as you gather more, yeah.
Okay. And another question from Dr. Sugimoto is about the conversion of the unit copy / microgram DNA to the pharmacologically meaningful unit copy per ml or milligram of tissue for CAR-T product modeling and simulation. Any comments on the units used for assays?
So it would, we use, I have to recall in terms of the units used, you would copy microgram per, I think, microgram per ml based on the copy number of your gene. I don't have the formula per se, but that's how we normalize in our, in our studies.
All right, and there were two additional questions that were put into the other chat, and one is, can you please comment on how a neutralizing antibody assay can be implemented for a target that is a membrane-bound ECD domain? Should we be thinking more in the context of blocking antibody assay than a neutralizing antibody assay?
If depends on if you're, if you could take advantage of your potency assay or express your cells in a cell line like a Jurkat cell line to express your CAR in a transmembrane fashion, that would be an easier implementation. But if you have reagents available for blocking assay, yes, of course, that would be easier to implement. I guess it would, what's the best way, easiest way to implement your assays right away?
All right, and one last question. I know we are at 12:30. Would you expect similar cellular kinetics in blood after local administration as in a solid tumor setting?
I suspect not, but I haven't seen the data to say either way, so I suspect not. Others can comment, but I think it'll be different.
All right, so we are at the 1.5 hour mark. I would like to thank all the speakers and also the participants who joined today and the questions that we received. You are welcome to send us more questions. We will be happy to answer. You can send it to me and I can triage them. We will be making this slide deck public. I don't know the logistics, but they will be provided. So again, please reach out to us, but we thank you today for joining us for this webinar. Thanks a lot. And thanks to the IQ organizers who helped us.
Yeah, thank you all.
Thank you all.
Have a good day.